Knudson was a physician who could be considered the Mendel of cancer genetics. Before his research, cancer was thought to be caused only by environmental exposure that caused sporadic mutations, as proto-oncogenes, genes involved in normal cell growth, were converted into oncogenes, genes that caused cancer. Knudson, however, hypothesized that cancer may be passed on genetically. This switched the focus onto the loss of function of tumor suppressor RB1 genes. Knudson studied retinoblastoma as an example, as symptoms for the hereditary form generally first appear in young children. Retinoblastoma is an eye cancer. The first sign is a brightness of the eye in dim light. Symptoms range from squinting and redness, even to blindness. If detected early, it can often be treated, but left untreated, the cancer can spread and be life-threatening. The tumor suppressor genes RB1 normally code for proteins PRB, whose function is to prevent the formation of tumors by acting as brakes on the cell cycle.
In simpler terms, imagine you're riding a bike down a hill. When you want to start slowing down, you can begin to pull on your right side handlebar, and even if the right side brake stops working, no problem. You still have your left side brake and your bike will come to a complete stop. But a problem arises when both your right and left brakes cease to function, because then there's no way of stopping your bike and you will continue barreling down the hill. This is the same premise as Knudson's two-hit hypothesis, where tumor suppressor genes in the body contain two copies of each coding sequence known as alleles. When one of these alleles experiences a loss of function mutation, the other copy remains dominant and is still enough to suppress tumor growth. However, when the loss of function mutation occurs in both copies of alleles, the tumor suppressor can no longer function, resulting in cancer.
So now let's discuss how Knudson created his two-hit hypothesis using statistics. Previous studies showed that 3 to 7 mutations in a single cell can lead to the development of common cancers. However, no studies have provided evidence that it only takes 2 mutational events in a single cell to trigger cancer development. Knudson began investigating 48 retinoblastoma cases recorded at the MD Anderson Hospital from 1944 to 1969. First, Knudson found that retinoblastoma occurs due to the loss of heterozygosity, where only homozygous recessive individuals develop tumors, because some patients containing the inherited gene did not develop symptoms for retinoblastoma, while their children did. Among Knudson's 48 cases, 23 were inherited and bilateral, while 25 were non-inherited and unilateral. These bilateral cases were seen to develop symptoms much faster than the unilateral cases. Knudson used this information to construct a plot of the fraction of cases not yet diagnosed as a function of age of children in months, showing that the fraction of bilateral cases yet to be diagnosed followed a one-hit linear curve, while unilateral cases yet to be diagnosed followed a two-hit exponential curve. Based on previous findings that retinoblastoma can be induced by somatic or germline mutations, his results supported the idea that since the disease is recessive, children with an inherited mutation in one of their RB alleles can develop it faster, as they only need one more somatic mutation. However, children who did not inherit a mutation in one of their RB alleles will take longer to develop the disease, as mutations are random. This led to Knudson's two-hit hypothesis. But for our case study, here's a quick breakdown of the two types of retinoblastoma observed by Knudson. The first type was familial, or bilateral, where individuals whose parents are carriers of the mutated RB gene only require a single mutation in the remaining normal allele in any retina cell to develop cancer.
The second type of retinoblastoma is sporadic retinoblastoma. This is when the child inherits two functional RB alleles or RB positive alleles. Therefore, the embryo is genetically normal. However, during retinal development, mutations occur in both alleles in a retinal cell. This cell rapidly divides to form a tumor. Here is a case study of sporadic retinoblastoma. Eliza is a retinoblastoma survivor. It all started when her family noticed that one of her eyes was becoming more and more lazy. After being referred to a specialist, she was diagnosed with retinoblastoma. Having routine eye tests allowed for the early detection of cancer before it started to spread. Unfortunately, her eye had to be removed within 2 weeks of her diagnosis to prevent the spread of cancer. Therefore, it is important to perform eye tests regularly to catch any early warning signs of retinoblastoma.
Knudson's research and his two-hit hypothesis for cancer development widened researchers' views from the previous model of cancer growth and provided a unifying model for understanding hereditary, also known as bilateral or familial retinoblastoma, as well as non-hereditary, also known as unilateral or sporadic retinoblastoma. The takeaway message of the Knudson two-hit hypothesis is that both copies of the tumor suppressor gene must be faulty for cancer to develop. Clinical relevance involves early detection of retinoblastoma. This is critical for both eyesight and survival prognoses, especially for bilateral retinoblastoma. In infant relatives of patients with retinoblastoma, regular ophthalmological tests are critical for early tumor diagnosis. Understanding and studying the mutations that produced a patient's retinoblastoma allows for reliable risk prediction for all their relatives. The end goal is to save more eyes, vision, and lives.